Hey everybody, welcome to Micro 2. This week we are going to be covering mycology, so I am going to just make sure I share my screen here so that we can go through the mycology PowerPoint. That should be in your lesson for week 1. So, all right. Get through the objectives. You can definitely read through those to know what to expect out of the chapter. But essentially, mycology is referencing fungus and molds. The basic general characteristics of fungus, again, is that they are eukaryotic organisms, meaning they do possess a nucleus and mitochondria. They also have these thick cell walls that are made of chitin. There are 2 groups of fungi in the clinical lab, and we're mostly concerned with yeast. And molds would be the other one, or you can commonly call them just fungi. But yeast typically will produce more of a moist or creamy type of colony, something more similar to bacteria. So when you do grow these on an agar plate, yeast are going to more resemble like a bacteria colony, whereas a mold is going to look kind of like what you would expect. Like when molds grow on your foods, you see that fluffy or woolly or cottony type of colony, and they'll look the same on agar when they grow. So there's what a yeast would look like. Again, much more similar to a bacteria colony where it's a little bit more moist, opaque in color. And then molds, again, they have all sorts of different textures. It just depends on what molds are growing. But words like powdery, velvety, woolly, those are common words to describe the texture of them. They're all different types of colors. They can be one color on the top, a different color on the bottom, that kind of thing. All right, so getting a little bit more into some terms that you are going to commonly hear throughout mycology. Hyphae is the basic structural unit of a mold. And the hyphae can either be considered septate hyphae or aseptate hyphae. So septate hyphae will basically have these distinct divisions in it, whereas aseptate does not have divisions. I'm just trying to skip so that you can see a picture. I'll go back, don't worry, but this is where you're seeing the division. This is septate hyphae. They have what we call these cross walls. Do you see where the arrows are? You can see others if you continue down the hyphae, which again, this is just the structural unit under a microscope of the mold. And so you can see those cross walls. That would be considered septate. Aseptate hyphae, if you look here, you don't see any of those cross walls, do you? So there's none of those. That's again, it means without septa, without those cross walls. So that is the basic structural unit of a mold, and mold will either be septate or aseptate. There is a division of molds that are common that are called Zygomycetes, that are very well known to be aseptate. Most others tend to be septate. Now, if you ever hear of the term mycelium, that is just referring to in the entire hyphae network altogether. So all of those hyphae together are called a mycelium, and this is ultimately what's going to be penetrating down into wherever it's growing on for nutrients so that it continues to grow. So there's the mycelia at work with all those little hyphaes, and again, it will dig down into get the nutrients, whether it maybe be from the air or down into the vegetation here, whatever it could be. All right, so with that being said, hyphae do take different shapes. I am not going to quiz you on the shapes here of these antler, racket, spiral, but I will quiz you on the rhizoid one. The first 3 here, you can just look at the pictures and you kind of get a sense of how they're named. I do not mention these again except here. The rhizoids, though, I do mention again, and I will test you on that. So rhizoid hyphae is referring to a root-like structure system. So if you look at this drawing up at the top here under rhizoids, you see those roots. It kind of looks like roots of a tree or a plant under the soil. So that's the rhizoids. That's the hyphae-shaped rhizoids that we're talking about here. So if you look at this fungus that is showing in the picture, this is a fungus called Rhizopus, and you see these little elements branching out here that look like those roots. That's the rhizoids. Now, you will eventually want to know there's 3 organisms we'll talk about that are separated between where the rhizoids are located. And so when we get into that, that's where rhizoids will become essential. So for right now, just keep in mind they're the root-like system, and so we'll come back to that here in a minute. Otherwise, reproduction of molds and yeast. So basically, there is an asexual reproductive process that is called Canidia, and Canidia are basically formed by cells that have these what we call fruiting structures. So they could either be a couple different types of fruiting structures. I will not test you on the terms Phyloconidia or Aneloconidia. These are just some of the different fruiting structures that might form, and then they will release out these conidia, and that's the asexual process. There's also a slightly different form called Arthroconidia. Most of our identification of mold is based on what the conidia looks like, and that's how we identify the mold. The main way that we identify and determine molds in the clinic lab is under the microscope. Microscopic identification is number one on how we figure out what mold is happening, what fungus is happening. And that's because they all look differently. So when we start getting into these fungi, it's going to be really crucial that you start to get familiar with the pictures, and so that you can identify the pictures. Especially at the very end of this quarter, we are going to have a lab practical in which you must be able to identify pictures of mycology. So that's where microscopic identification becomes really crucial, and a lot of the microscopic identification is based on how these conidia look. They will look different depending on each organism. So this is just showing you those fruiting structures. This one is the Phyloconidia. It's described here as being more of a vase-like structure. You can see it's kind of that vase, and then the conidia is at the top coming out. There's the hyphae, which is the main structural unit, and then it develops up into that vase-like system with the asexual reproduction. This is the Arthroconidia, which was called more of a ring-like fruiting structure. So this is just showing all of those there together. They kind of look like little rectangles here. Again, these last two slides are just informational. I will not test you on these two slides. Okay, so identification of molds, like we said, microscopic is the #1 way to identify molds, still is. Other things that go into identifying molds is their growth rate. Molds are very slow growers. If you think about how long it sometimes takes to grow on food, it can take quite a while to grow on food. Now, granted, maybe your food was sitting in the grocery store before you bought it for a while. That's why it grew more quickly to you, but it still takes quite a while. It's not overnight. It usually takes days and days and days before molds grow. Also, the colony, as I mentioned earlier, different characteristics of the texture of the colony, the way it grows on the agar, the color, that all can also go into identification of it. But again, the microscope is going to be your #1 friend for identifying. So we will categorize our fungi into different families here, basically, Zygomycetes, Ascomycetes, Basidiomycetes, and Zygomycetes. And within each of those, they will have different organisms that we'll discuss the clinically important organisms within each. I don't care about all the others that are not clinically important. So we're going to start with the family Zygomycetes. Zygomycetes, again, were the family that was more aseptate, that did not have those cross walls. And they will perform an asexual reproduction by producing sporangiospores. So sporangia is the fruit structure in this case, and then that produces spores from that. They do have a sexual reproductive process as well through a term called zygospores. Again, this part is just informational. There are 3 clinically important organisms within this family. It's Rhizopus, Mucor, and Absidia. These are the 3 that I need you to differentiate based on the rhizoids. So based on how those rhizoids are, those root-like rhizoid structure with each of these 3. So this is just showing a picture of that sporangia with the sporangiospores inside of it for asexual reproduction. Here is what it looks like under a microscope. Aren't they pretty? Molds are so beautiful under microscopes. Oh my God, they look like flowers a lot of times. They're nasty things, but they look like flowers under a microscope. And then this is just showing you the sexual reproduction process with a zygospore. It's just informational, but it is sexual process, so it does involve a male, female, that kind of thing, but whatever. All right, so like I said, we need to differentiate those 3 clinically important organisms, Mucor, Rhizopus, and Absidia, based on where the rhizoids are. So here's the definition of a rhizoid. It's that root-like structure. And so these are the 3 you need to remember with that. I'm going to come back to those slides, but since we're on the topic, I want to get to it. Rhizopus has the rhizoid root-like structure at the very bottom, the base. So here's Rhizopus, this little thing that looks like a flower with Sporangiospore at the top. That's that fruiting structure. It contains all those little spores inside of it. So this is A and B are Rhizopus. C is showing you the rhizoids at the very bottom of it, that root-like structure. So know that Rhizopus contains its rhizoids at the base or bottom. Absidia will have rhizoids between the two sporangiophores. So in this case, you see there's two sporangiophores. One is lettered A, and then there's another one here. And right between those two is the root-like structure lettered B there. So it is in between. It's not the best picture, but it is in between those two sporangiophores. So know that for Absidia, it occurs between. And then Mucor has no rhizoids. It's belonging in the Zygomycete category, but it does not possess any rhizoids, so keep that in mind. That's all you need to know on Rhizopus, Absidia, and Mucor, is that they belong to Zygomycetes, which are aseptate, and you need to just know where those rhizoid structures are. And in the case of Mucor, it doesn't have any. All right, now let's go back to the slide I skipped. So what do these three cause for disease? Overall, fungal infections typically affect people that are immunocompromised. We are exposed to fungus on a daily basis all around us in the environment. And we primarily contract molds and fungus through inhalation because they all produce spores, which are in the air. So you can find fungus everywhere in the soil, in vegetables, everywhere. So you're constantly being exposed to them. But because you have an intact immune system that works pretty well, you fight it off. It doesn't cause any problems to you. Those that are immunocompromised are the ones that usually suffer with a fungal infection because they can't fight it off. So they would be people that are elderly, newborns maybe, that just don't have a great immune system. People that are HIV positive, that have leukemia, burns victims, diabetes, people who have really complicated diabetes, things like that are people more at risk. So as it says here, Zygomycetes tend to infect those immunocompromised patients. Again, acquired by inhalation of spores. When it does become a fungal infection, it's very serious. It's not something we take lightly. Because they're already immunocompromised and then they're getting into their lungs and it spreads to other organs, it's a nasty, nasty infection. It's not easy to get rid of. So it will become rapidly progressive. Okay, moving on to our next little family is Ascomycetes. These are Septate Hyphae. Again, they reproduce asexually by forming Candida, which we will see over and over. They reproduce sexually by producing ascospores, which is why they're called Ascomycetes, for the ascospores. The ones that are clinically important in the Ascomycota category is Microsporum and Trichophyton, and then another one that I can never say the darn name, but I believe it's Pseudolascaria boydii. So those are the three clinically important ones here. All right, in Basidiomycetes, the other category, again, these produce Basidiospores, which is why they're called Basidiomycetes. The only clinically important one here that I need you to remember is Cryptococcus neoformans. That's the one that's really big, especially with HIV patients. It's commonly found. And then Deuteromycetes basically doesn't have a sexual reproductive process. It's just an asexual Canidia. And then this can cause loss. Basically, this has the biggest amount of organisms in this family. All of the organisms in this family can cause either cutaneous disease, subcutaneous disease, or systemic disease, which is the worst category of all. So this will contain the majority of the molds that we talk about. All right, so now that we've kind of gone over like the whole, family overview or whatever you want to call it, now we can categorize molds based on disease. So we have our cutaneous mycoses. Mycoses just means a fungal disease. So we have cutaneous mycoses, subcutaneous, systemic, and then those that are just opportunistic. So this is the main way that we are going to learn our molds, is based on this type of categorization. Because as far as I'm concerned as a clinical laboratorian, I'm worried about the clinical disease. I'm worried about what's happening with the patient. I'm not as much into the science of Deuteromycetes. I'm more worried about what kind of disease here are we happening. Okay, if it's a cutaneous infection, I know it could be one of these molds then, that kind of thing. So I'm more worried about this kind of categorization. All right, so we're going to start with the cutaneous molds. These are going to be basically infections that either involve your hair, your skin, or your nails. So those are all cutaneous levels or superficial levels. So think of things like ring-like patches on the skin, itching, thickened, discolored nails, brittle, broken hair, those kind of things. And it just stays at the superficial cutaneous level. It does not go any deeper. So typically these are called dermatophytes, derma for cutaneous. So they are going to cause things that are called tinea, tinea nigra, or piedra. I don't know why I tried to say an R, rolling R with that, tinea nigra. So basically, tinea just means a fungal infection. All right, so here's just an example of your skin layers, and we are staying right up here at the epidermis level only, at the very cutaneous top part of this. Well, I should say the dermis and epidermis, not anywhere beneath that. Okay, so dermatophytes, again, the main dermatophytes that we're worried about here is Trichophyton, Microsporum, and Epidermophyton. The epidermal one is nice because the name is skin right in it. And here's all the different types of basically mycoses that can happen at the cutaneous levels and what part of the body they go with. So tinea, again, is referring to the fungal infection. And the second type of word refers to where on the body it's occurring. So tinea capitis, cap is head. Tinea fibrosa is also referring to a distinct part of the head. Tinea barbae is for beard. You think about barber, like a barber that trims the beard. Tinea corporis, corpus just means the whole body. So it can be somewhere on the body in general, cutaneous-wise. Tinea, I can never say this, menuum is hand. Unguium is nails. Cruris is groin. Pedis, of course, is feet. And then we have other ones on the body called imbricate. So lots of different cutaneous forms, and then the words tend to tell you where on the body it's happening. All right, so we're going to start with some of our dermatophytes here. The first one we're going to start with is called Malassezia furfur. This is actually a yeast, not a mold, but a yeast. And this will produce lesions that basically have pigmentation to them. We call them often versicolor because they have different colored pigmentations. And typically this happens on the chest, the trunk, and the abdomen. It's been kind of linked as a cause of dandruff as well. But the big thing with this, again, because remember, microscopic identification is #1 key to anything, it will have what they call a spaghetti and meatballs appearance under the microscope. So there, it's supposed to be showing you the spaghetti meatballs. Now, you're probably thinking, that does not look like spaghetti meatballs, but keep in mind, it's a lab. It's never like picture perfect friendly, but I think down in the right-hand corner, it looks more like the spaghetti meatball mixture because you can see the little round Candida for the meatballs and then the little hyphae for the spaghetti more. All right, piedra forte causes what we call black piedra, which are basically hard black nodules around the scalp hair. So this is more seen in Africa, Asia, and Latin America. So it's showing you these nodules around the scalp hair close up under the microscope. Trichosporum causes oftentimes what we call white piedra on the hair shaft. Again, South America, East, and Pacific. This is showing you Trichosporum under the microscope. You do not need to know this picture here. I would know the Malassezia furfur one, but I would not need to worry about the Trichosporum here. Hortea werneckii is also called Phaeoanulomyces, which basically causes tinea nigra, which is a brown to black pigmentation on your palms and feet. So oftentimes we do skin scrapings to identify dermatophytes in KOH prep. So KOH is potassium hydroxide. It's basically you add the skin scraping onto a microscope slide, you put KOH on there, a couple drops, and it dissolves any other cellular elements, and that way if there is mold or yeast present, they can come through better and you can see them better because it gets rid of all the other crap. So oftentimes we are looking for fungal elements with KOH because it really lets them shine through. So this is going to show more yeast-like colonies. Again, yeasts tend to be more oval in shape. They're refractile under the microscope. When you focus it up and down, they're not as round as a red cell. They're easily confused, but they're a little bit more oval in shape. So this is, again, just talking about that potassium hydroxide preparation, something very commonly asked for whenever there's a skin scraping done. So again, it dissolves the skin cells, but not the fungal cells, so that way anything yeast or mold-wise that's present will shine through. All right, Trichophyton is another dermatophyte that involves the hair, skin, and nails. This has more of a club-shaped macroconidia with 3 to 8 septa overall. Oops, sorry. And so within Trichophyton, there's several different organisms. We have Trichophyton rubrum. Trichophyton rubrum, the biggest characteristic with this is the color it takes on the agar plate for the colony. So on the top, it's more of a white to reddish kind of cottony look to it. On the bottom, it's a very classic, characteristic cherry red color. That's kind of what indicates the rubrum part, is that bright cherry red color. It's bolded because it's a huge characteristic of Trichophyton rubrum. Under the microscope, it will have more cigar-shaped macroconidia and tear-shaped microconidia that are said to look like birds on a fence. So this is just showing you the white colony on top, and you reverse it over, it's that cherry red, and they're showing you the little birds on a fence. Isn't that cute? So cute. Oh, okay. All right, so again, causing a dermatophyte type infection, so it's causing infection of the skin and nails. Commonly causes with tinea corporis, which is all over the body. Trichophyton mentogrophites is another skin scalp lesion. A lot of times goes with tinea pedis. White, cotton, yellow colonies, and then the reverse side is more of a rose brown. The big characteristic with this one is the microconidia are said to be more like grape-like clusters when you see it under the microscope. So there's letter A showing a little bit more of the grape clusters. Letter B is showing you the macroconidia in a cigar shape, and that's mentioned here at the bottom. Here is another picture of all the little grapes. This one doesn't show the macroconidia so much. This isn't the best picture, but yeah. Trichophyton tonsurans causes tinea capitis, which is on the head. This will have a very unique look on the colony morphology as well, in that it has radial folds and a crater-like depression in the center on the colony. So there you're seeing the radial folds there, and you see in the center it's got that depression. And you can see overall how it looks on the right. Here's what it looks like under the microscope. You do not need to know this picture. I won't make you identify the microscopic image of this one. Trichophyton varicosum, more lesions. It's a huge cause of ringworm, especially in cattle. And these are button-shaped with a white raised center for the auger. So there you can see it on the cattle. And there you can see the button shape with the raised center. It is a raised center. You can't see it's raised here, but it is. And then as far as under the microscope, it produces chlamydospores in chains. Again, do not need you to know this picture at all, nor this one. Don't worry about these two pictures. Just know that, I know this seems like a lot, you're probably already feeling overwhelmed with all these fungal elements. Flashcards are always helpful with these kind of things, but you do not necessarily have to remember every single little thing about these. For instance, you could just try to remember what's in bold, or if anything really stood out on each one, and just try to whittle it down to the basics. All right, Microsporum also causes more dermatophyte type infections. So these have more large macroconidia with usually more than 4 septa, and then smaller microconidia. There is one that's called Microsporum adoni, I can't even say the word, that one that causes tinea capitis. I don't test you on that word. Don't worry about that organism mentioned there. It's just information, but you do definitely need to know Microsporum canis. I for sure test you on this. I for sure test you on the picture of this. So Microsporum canis, just as it sounds with dogs, it's a huge, actually it's the #1 cause of ringworm in dogs and cats. So it's the most common cause of ringworm. And typically when humans get ringworm, it's from handling infected animals. So when you grow this out on the auger plate, it's got kind of like a white, silky top to it. The bottom tends to be more lemon yellow. I don't really care about the colony morphology. What I care about is what's in bold here under the microscope. It has very much got these spindle-shaped macroconidia, which means that they come to a point at the ends. So do you see how they come to a point down there? That is so key with Microsporum canis. So you see the septa, there's 1, 2, 3, 4, 5, these 6 septa there, and I think there's supposed to be 4 to 8, yeah. And then it's got these pointier curved ends at the end. That is Microsporum canis through and through, definitely should know that. Here's another picture without the dye to it, but you can definitely still see all the septa and the more pointy curved ends there. Microsporum gypsum, I would know this one too. Doesn't cause that much infection, but just in case you get asked, its macroconidia under the microscope have more rounded ends. Still has about 3 to 9 septa, but a lot more rounded ends. These are not quite the point that it comes down to with the other one. Definitely know Microsporum canis over this one, but if you want to know both, I would. And then here it is again, not quite that curved down point. Epidermophyton flocosum, definitely would know this one as well. This causes tinea crurus, which is the groin, and tinea pedis for the feet. This one, under the microscope, it has a club-shaped, or they call it beaver tail macroconidia. Hugely important that you remember this, club-shaped or beaver tail macroconidia look here for Epidermophyton flocosum. Okay, so that was all the cutaneous dermatophytes. I mean, that's a freaking lot. All right, we only have one main subcutaneous mycoses that I need you to remember, thankfully. So now we're just going to the subcutaneous infection, which involves the deeper skin layers. And so the only one I need you to know out of this is Sporothrix shanki at the very bottom. Sporothrix shanki will be mentioned later again because it does cause a systemic infection, but it starts out as a subcutaneous infection. So again, we're going to the subcutaneous layer at the very bottom there. So Sporothrix shanki is found on vegetation, commonly with plants, and this is usually passed through when people are tending to their gardens and like pruning their plants. It's commonly called a rose gardener's disease. So they get it from being pricked with thorns on the roses, and then it will grow into a subcutaneous type infection, and then it can go into a systemic infection. Now, this has both yeast and mold states, which we'll talk about that later, but yeasts are more rare. We don't see those as much, but you should definitely know what the molds look like under the microscope. And they are single-celled conidia that have little clusters at the tips. So letter B is the mold that I need you to know. So if you look here, they get like kind of look like little flowers there at the tips, the longer ones. I know they're a little clustered. Here's a great example of what it looks like. So definitely know that picture and know all about Sporothrix shanki. All right, moving on to our opportunistic fungal infections. So these are things that will take advantage if they can. We have Aspergillus, Fusarium, Geotrichum, and Penicillium. So Aspergillus, hugely found outside in the soil. Again, you get it by inhaling conidia, typically more with immunocompromised patients. This isn't more of an opportunistic one. So it will only cause infection if it really has the opportunity to. So that patient has to be really pretty down on their immune system. So Aspergillus fumigatus is a very common one out of the Aspergillus family. This one has Septate Hyphae. It has what they call a foot cell at the base of the conidiophores. It basically looks like a foot. To me, I don't know. I don't think it looks like a foot, but whatever. It's pretty. I think it's pretty. So this is Aspergillus fumigatus. You can see the spores that come up off the sporangiophore at the top there. So that produces, yeah. So make sure you're familiar with the way this looks. Aspergillus fumigatus, the other thing you should know is this is the one that they often call farmer's lung. Because it's so found in the soil, and as farmers are out tending to their fields and turning up the soil, if they're immunocompromised, they can breathe this in and become sick with like a pneumonia from it. So oftentimes causes farmer's lung. Aspergillus flavus also has a spherical vessel that has all the little conidiophores and conidia coming off of it. It kind of looks like sunflowers to me when you look at them. Aspergillus niger is called niger because it's more darkly pigmented spores. When it grows on an agar plate, it has a black dotted surface as well to it. The big thing to identify Aspergillus niger is it's a very large spherical vessel, much more larger than the other two, and its entire surface is covered in spores. So much more bigger than the other two Aspergillus. So out of the Aspergillus, I would definitely remember Aspergillus fumigatus and Aspergillus niger. All right, Fusarium is our next opportunistic mycoses. This has a very unique shape. This one's a lot easier to remember than the others in that it's a sickle cell looking or boat-shaped macroconidia. So it looks like sickle cells if you think about it, just a different color. So that is Fusarium. Definitely know that. And there it is again. These are a little harder to pick up, but they look like a bunch of bananas because they're yellow. Okay. Geotrichum, it causes pulmonary disease in immunocompromised. This has kind of a flatter white to cream suede type texture to its colony. I don't really care about the colony morphology on this. I more care about what it looks like under here. So you can see it's very rectangular looking and definitely chains. So Geotrichum tends to chain and be very rectangular. This is a really good picture of it. Look at how much it chains. It almost looks like little grandpa's rods, doesn't it, chaining together? Okay, and Penicillium is the last of our opportunistic ones. This definitely has what we call brush-like conidiophores, or you can call it a paintbrush look. And it definitely looks like a little paintbrush there, right? So that should be very identifiable to you. Definitely remember Penicillium. Penicillium was basically the fungus that made the antibiotic penicillin. They made it off of this fungus. It's something that you can commonly find growing different places. Again, it's opportunistic though, so most people's immune system are good. They just fight it off, but there's a little paintbrush look on it, so definitely remember that. All right, and then we have a category that we call Dimorphic. Basically, this is fungus that are darkly pigmented is what it means by Dimorphic. So they have a melanin production. So most others we always call hyaline because they're non-pigmented. These ones have a pigment to them. They have a melanin production. And the one that you need to know out of here is Alternaria. So this has definitely got a pigment to it, so it's in that Dimorphic category. It does cause cutaneous infections along with some other stuff. Its colonies are gray to black because, again, it's pigmented. As far as under the microscope goes, it has this very unique look, and they call it an acropetal, in which it's kind of elliptic or oval, and then it tapers, kind of like almost like the Microsporum canis, but just a slightly different way. It's a little bit more rounded at the one end, and then it's definitely got this pigment. It has this brown pigment to it, whereas Microsporum canis, you have to stain it to get it to look like a different color. So I would make sure you are aware of Alternaria. All right, and then finally, our last category by diseases is the big bad boys, our systemic fungal infections. There are 5 of them that cause systemic infections, meaning they can cause infection throughout the body. We already mentioned the Sporangiospore because we talked about the subcutaneous, but I said in there it can become systemic, so it's mentioned here, and then we have 4 others. All 5 of these are what we consider dimorphic fungi. You see that's in bold, so we will discuss that along with each of these 5. So a dimorphic fungus means that it can be both a mold and a yeast. Typically, fungus fits into just one category or the other. Either they're a yeast or they're a mold. These are the ones that can exist as both dependent on temperature. So temperature will decide what phase they're in. So the mold form will occur at more the room temperature, 25 to 30 degrees, whereas the yeast stage occurs at body temp, which is 35 to 37 degrees. The way that I always keep this straight is I think about my food sits on my counter. Like if your bread sits on your counter, it forms mold at a room temp, right? So I always just think of it that way. These are very much virulent, meaning they are quick to cause disease when they can in susceptible hosts, and they again become systemic. So there's the 5 summarized in a chart out of your book. I just thought it was a nice chart, and the way that they look on the colony and then the way they look under the microscope. If you want to read through that, you definitely can. Here is what I was talking about for the temperature with the yeast form. This one over here I think was more, I thought it was more of the mold form, but anyway. All right, and then here is what the diseases that they cause. So Blastomyces dermatitis is often found in the Mississippi and Ohio River valleys, and again, because it's systemic, it's going to involve more than just one area. It pretty much usually always starts with lungs because you inhale these as spores. So they'll start with lung, and then like blastomyces will go to skin, bone, and then the way that it looks is described here. So the big thing with Blastomyces dermatitis is when it's in the yeast stage, it's a very large, thick-walled yeast. You can usually see the lining of the walls. So I think I have a picture of that coming up. Histoplasma capsulatum is seen in the Ohio, Missouri, and Mississippi River valleys, also seen with bird and bat guano, so bird and bat poop. Again, primary lung, and then it goes into a disseminated disease. Coccidioides immitis is seen down where I live. This is something that's commonly seen down here in southwest U.S., down in Arizona. And so this is the valley fever. They call it a valley fever down here. So again, it starts as lung and progresses. And then you have Paracoccidioides brasiliensis. Think of Brazil for South America, and that's where it usually is stationed at. Okay, so this just goes back through that. That was out of your book. So again, blastomyces dermatitis. The disease is actually called blastomycosis. So again, this is the Ohio and Mississippi River valleys, like it said here. So in North America, starts respiratory, lungs, and then spreads to bone, tissue, and skin. Again, it can occur in both yeast and mold stage because it's dimorphic. The one stage that I want you to remember with blastomyces is the yeast stage. And that's that thick-walled, large yeast look to it. So there is the yeast stage, and you can see the two lines where the wall is. That's the thick wall. You should never really see that wall lining like that. It's so thick. Here it's even better picture. You can definitely see the thickness to that wall. And yeast always bud to reproduce, and that's what these are doing here is they're budding. And then here's the mold stage. They tend to look like little lollipops. I will not quiz you on the mold stage. I just want you to know the yeast stage. And there it is again. All right. Coccidioides immitis is the valley fever I said. That's down here in Arizona, southwest U.S., Mexico. Again, respiratory tract spreads to lots of other organs, including bone and skin and tissue. The yeast is more of a non-budding form that you do not have to worry about. The mold is the one that I want you to know how to identify. The big thing with coccidioides is it's easily transmissible to others in the lab when we're working with it. It's so huge to make sure you only work with this under at least a level 2 hood. Very, very infectious of all the fungi, so it's easily to pick up by other lab workers and actually become sick with it. So if you're not working with this under level 2 hood, you're going to get sick. So that's where the caution comes in. Under the microscope, it has this rectangular or barrel shape to its look. That's the yeast stage. Again, you don't need to know the yeast stage, but I would know this mold stage of coccidioides. So there it's showing you kind of the rectangle or barrel look to it. And there it is again. So definitely know coccidioides molds. All right, histoplasma capsulatum causes histoplasmosis. Starts kind of like a granulomatous infection that begins in the lung and then quickly continues from there. Found in North America river basins. The one that I want you to know for the stage on histoplasma is the mold stage. It's got what we call a round tuberculated macroconidia, which means the macroconidia looks very rough on its edges. So this is just what it looks like in tissue. You can ignore this. But here it is. That's what you need to know. So you can see it's this round conidia, and then it's tuberculated or rough looking, right? A little spiky. That's what's key to identifying histoplasma. There it is again. You'd have to kind of really squint, but you can see the spikiness. And then Paracoccidioides brasiliensis. This is the one that's in South America. It's named Brazil. Again, this systemic disease. I mean, you basically can leave it as systemic disease. And the look that I want you to know of Paracoccidioides is the yeast look. It has a very characteristic look that we call a mariner's wheel. So when you think of a ship, like a pirate ship, and how they used to steer the pirate ship with the wheel, that's a mariner's wheel where it has those spokes. And so that's kind of what it looks like here. It'll have these budding yeast cells coming out of it to make it look like that mariner's wheel of a pirate ship. So it's showing you it there. Now, it's not picture perfect like what you're thinking in your head, but it's got little spokes coming out of it. That's the look that you should know for Paracoccidioides. There it is again, the little mariner's wheel. There's the mold stage. I do not need you to know the mold stage of this one. All right, so this is just showing you where you can see predominantly any of these systemic organisms occurring. Again, they all start respiratory because you're breathing it in, and then they can quickly disseminate to some part of your body Most of the time it's involving your skin. Okay, moving on to yeast. We haven't really talked much about yeast. We talked mostly molds. Now we've talked dimorphic fungi, and now we'll get into yeast. Yeast will produce asexually by budding, which we call Blastoconidia formation. So that's what the picture is showing you, how they begin to bud, and then they will separate. So it's a mother and daughter cell upon separation. So there it's showing the budding in a picture format. The yeasts that are considered clinically significant in the lab is Candida, Cryptococcus, Trichosporin, and Malassezia. So we've already talked about Malassezia furfur with the cutaneous infections. That's the one that's the spaghetti and meatballs. Trichosporin was mentioned in the cutaneous as well. I don't quiz you a lot on Trichosporin. And then we have Candida and Cryptococcus left to discuss mainly. So when you identify yeast, typically we're not going to always identify yeast as just anything. We're especially, though, going to identify yeast if it comes from something that's supposed to be sterile. So if it's coming and growing up with cerebrospinal fluid, because it can cause meningitis, if it's coming through blood cultures, or if it's seen in urine, then we want to definitely identify what yeast it is. We want to also identify the yeast if it's coming from somebody that's known to be very seriously ill, because, again, it's going to really cause a problem for those immunocompromised patients. If we're thinking of sputum, like a respiratory secretion, then you might want to look especially for Cryptococcus neoformans, that kind of thing. So let's look at Candida. Candida is the most frequently, out of all fungal infections, out of all molds and yeasts, Candida is the guy you're going to see if you see anything at all. Candida albicans is the most common out of the Candida. So this will cause disease like thrush, which involves like the white patches in your mouth, meningitis, pulmonary infections, eye infections, endocarditis, and you could probably keep going with that list. So Candida, how we identify, again, they tend to look more like bacterial colonies on an agar plate. They'll kind of have a creamy consistency. Sometimes they can be a little drier, but white colonies. The neat thing is with Candida albicans, because that's the most common, one of the ways we can identify it, just because it's so commonly seen in the lab, is through what we call a germ tube test. A germ tube is essentially the first start of hyphae formation. So when it's just going to first start to form hyphaes. It doesn't have any constriction at the base of it, and Candida albicans is definitely known to produce these little germ tubes. They kind of look like sperm. When you see the picture, you're going to think it looks like sperm. You can also grow yeast on tween agar, just so you know. But there's the little germ tubes there. Like I said, they look kind of like almost like little sperms. But that will give you a positive for Candida albicans. So know what a germ tube looks like and know that it's to tell you you have Candida albicans present. There it is again. Again, there's no constriction at the base here. It just goes straight into the hyphae formation. So it's just starting to form hyphae. You also have pseudogerm tubes, which are not true germ tubes. They look like it, but they're not. And the difference is they do have a constriction at the base. So this is showing you a germ tube with no constriction, and then you have pseudohyphae that has constrictions. I do not test you on pseudohyphae. It's just a mention of it. I just want you to know what a germ tube looks like, and Candida albicans is what it's testing for. All right, and then Cryptococcus, especially Cryptococcus neoformans, is the next yeast we want to discuss. Commonly linked with pigeons. It will cause a lot of problems with AIDS patients. I think I mentioned this way back at the beginning when I first said the name Cryptococcus neoformans, that it was heavily linked with HIV or AIDS patients. Causes meningitis, pneumonia, septicemia, all very, very serious illnesses for AIDS patients when their immune system is already down. So again, it is a yeast, so it'll kind of have that yeast-like look on the agar plate. The biggest way to detect this, now I heard that they don't use this very much anymore, but I want you to still know this because your boards can ask you questions on it. And this is called an India ink stain. Cryptococcus neoformans basically has a capsule to it. And so when you go to stain the sample with this India ink stain, it will stain everything except the capsule because it cannot penetrate inside of it. So when you look at it under the microscope, everything will look basically like a brown color, except Cryptococcus neoformans. It will shine kind of like a halo almost because it's perfectly clear, and that will tell you, yes, you have Cryptococcus neoformans present. There is now an actual latex agglutination test you can do with it, but it used to be that they always did this India ink stain. I don't know if they're doing it that commonly anymore, but I would still know about it. So here's what it looks like. So you can see how everything kind of stained a brownish color except the Cryptococcus neoformans yeast. That has a capsule, so the stain couldn't get in there, so it left it looking clear and looks like a halo. All right, so this is just talking about yeast identification. If it does have a germ tube and it grew at 42, it's probably in Candida albicans. If it did not have a germ tube and it grew on, whatever. You do not need to know this flow chart. We just love to flow chart everything in the lab, so I wanted to show you how you can possibly flow chart a yeast instead of a bacteria. Again, I do not expect you to know all this. This is a lot of extra information. And then pneumocystis. Pneumocystis used to be part of the parasites when we discussed parasites. Now they have decided to put it in the fungus category. To me, I'm still so used to thinking of it as a parasite that I kind of forget that they put it in the fungal category now. But essentially, pneumocystis is, again, a big problem with AIDS patients, immunocompromised in general, but AIDS patients particularly. Pneumocystis carinii was the big one with AIDS patients. So just recognize that pneumocystis carinii, very commonly found with rats and seen as an opportunistic infection in AIDS patients. Pneumocystis urevechii also causes infection a lot of times with younger children. So again, it's going to be more of a lung type infection, difficulty breathing, that kind of thing. So this is just talking through the stages of pneumocystis. I am not going to make you remember this or know this, so you can just ignore this slide. And then you do not need to know the pictures either. I don't care about the pictures on this one. And then again, just more flow charts to kind of give you information on how we can flow chart like anything. So it's just an example. Remember, calcifluoro white, do you see that mentioned here? We learned about that last quarter when we learned about different stains. Remember, calcifluoro white is specifically targeting the cell walls of fungus. So it is a fluorescent stain that's perfect for looking for fungus because it stains the cell walls. It binds the cell walls. So just keep in mind that works with mycology. And then as far as different media, we have lots of different media that we've never talked about that are used to grow molds. I do not make you memorize the media for molds. The only one I think you need to remember is tween 80 for Candida. But you do not have to worry too much about media with these. I don't test you on them like I did with the bacterial media. And yep, this is just more media information. Again, we do have antifungal treatments, just kind of like we have antibiotics, we have antivirals if we need to. I do not make you memorize these either. This is just information at this point. Okay, I know, my gosh. It is a ton of information for starting week 1, but I think I've tried to be as clear as possible what you do need to know out of this. Start getting familiar with the picture, start remembering any little key tidbits. Take the information out, put it in flashcards, write it out somewhere else, whatever you need to do. By no means do I expect you to memorize 138 slides. Oh my God, that'd be crazy. But if you whittle it down, it's a manageable amount. So that's it. If you guys have any questions, as always, please feel free to email me or give me a call, whatever you need. Otherwise, have a great week.